Hello, everyone. Welcome to today's lunch hour lecture. We are very fortunate to have here today Professor Charles Swanton from the UCL Cancer Institute. So that's where he works. He was just explaining to me how lovely this building is.
It's actually the Francis Crick Institute. So that's the Francis Crick Institute, UCL Cancer Institute, just across the road from here.
You didn't tell me
no,
this.
Two institutes I work in. Sorry, I should have explained.
Okay. So I have to go there to check. So he's a fellow of the European Academy of Cancer Sciences and has published over 120 articles in journals such as Nature, Nature Genetics, and Cancer Discovery. I think that his research is key to what we know today about genomic diversity within cancers. You can use his Twitter to tweet about this lecture, or the Twitter feed for UCL lunch hour lectures at UCL LHL. The Slido code event is 2074. After Charlie's lecture, we are going to be taking questions from this room and also from the online audience. So now with Charlie, thank you.
Thank you very much. Thank you all for coming. I'm going to talk today about work that we're doing at University College London, Cancer Institute, and the Francis Crick Institute, and within my work as a medical oncologist treating lung cancer. So what is cancer? I mean, it's an easy question to ask and a very difficult question to answer. I'm going to try and give you a sort of overview of the complexity of cancer today and how cancers evolve within a single host over that patient's life history. So cancers derive from a single cell. Cancers derive from a single cell, usually due to a point mutation in DNA that triggers expansion of that first cell. Now that first cell expands and it invades through its basement membrane. It becomes resistant to cell death. And ultimately, it spreads around the body to sites like the brain, the liver, the other lung, or the bone, and causes death through a myriad of different ways. We're not entirely sure how, very often. It's an unanswered question, something I'm very interested in. But in essence, cancers are driven very early in their evolution through the selection of one or more driver genes. Now, those driver genes may involve genes which we can target in the clinic. So targetable oncogenes, we call them. These change the behavior of the cell. They enable the cell to proliferate faster. They gain an additional advantage over their surrounding environment. And ultimately, they can then invade through the basement membrane and develop new characteristics that allow them to survive in the bloodstream and colonize new sites of disease. Now, the problem is, in the clinic, that we can target many of these cancer driver genes. But the difficulty is that resistance is acquired in every tumor in a very short space of time. Within 3 to 12 months, almost all patients have developed resistance to therapy, and in the metastatic setting, die of their disease. And it's very, very hard to cure a tumor that has spread beyond its primary site for the most part, with some exceptions like germ cell tumors, seminomas, testicular cancer, and what have you. So the problem that I face in the clinic treating cancers of the lung, the breast, the ovary, etc., is that the drugs that we use are incredibly expensive. And as I've just said to you, the benefits from treatment are relatively limited. So over the course of the last 12 years, if you look at the cancer drug approvals in the FDA in the United States, 71 anti-cancer drugs were approved. The median overall survival benefit was a paltry 2.1 months. And the cost of therapy is immense, $10,000 US per month, translating to almost millions of dollars per life year saved. Now, in an economy like ours, where we're spending 120 billion pounds a year more than we're earning, this is just unsustainable. So what are we going to do that can better this? And why is cancer the emperor of all maladies? Why is cancer so hard to treat? When I was at medical school, we were taught, in fact, right the way through my oncology training for the last, between, I guess, 2002, 2008, I was taught that cancers evolve in a linear fashion. So you get one point mutation that leads to expansion of a group of cells. And then you get another mutation, and that mutation gives that new cell an additional advantage that then expands that group of cells, such that you get sequential ordered linear evolution and expansions of clones of cells that would lead or force in that tumour homogeneity, and by that I mean every cancer cell is the same. So no matter where you take a biopsy in a patient or a patient's disease, that biopsy will give you the same result, whether you take it from the breast, the chest wall, the lung, the liver, or the brain. You'll essentially get the same result. Now, this sort of linear path to evolution just didn't make sense. At the bottom here, which is cut off from the screen, it says if linear evolution is true, why are cancer cures so evasive? And why is cancer so hard to treat? So that sort of question formed the framework of the first big experiment we did in this field about 6 or so years ago, where we took a patient who had advanced kidney cancer. You can see the primary renal tumour up there, the kidney cancer in the left-hand corner, a large 10 by 5 by 5 centimetre tumour mass that was resected at surgery. And we took multiple biopsies from that primary tumour and the patient's metastatic site. So the patient had a chest wall metastasis that had spread from the primary right renal cell carcinoma, as well as a perinephric cancer metastasis from the primary tumour. And we sequenced the whole coding genome of that tumour, 22,000 genes, about, I guess, 300 million base pairs we sequenced in very deep detail to understand whether all of the biopsies will give you the same results when you sequenced every tumour genome in each of those biopsies, or whether they give you very different results. On the right-hand side, you see the mutations that we found in each tumour region to ask that question, whether tumours are evolving in a linear manner or not. And what we found is this, that essentially in those 9 regions that you can see in the top right-hand corner, those are the mutations in red that are shared in every tumour region. I won't show you the gene names, they're largely irrelevant. But then when we looked at all 9 regions and piled them up, we found some mutations that were shared in all of the primary regions, but not the tumour metastasis, some mutations that were just present in the metastasis, and some mutations that were private, specific to individual tumour regions. Now, the problem for a clinician is if we generally rely on one biopsy, what do we treat? We've got point mutations here that could be driver genes that are present, let's say here, just in region 4 of the primary tumour, but nowhere else. But if I just take that region here and no other regions, and I treat the patient based on that one point mutation, that's clearly not going to have much effect if that mutation isn't shared across every region of the tumour. So we formed this model based on work from us and others in the field that tumours are evolving in a branched manner. This tumour is not evolving in a linear manner at all. There are multiple different subclones in this patient's tumour, multiple different cells that are surviving independently from each other that share a common root, a common trunk. And in this case, the driver mutation that is present in every tumour cell is this mutation called VHL that you can see in the trunk of this patient's evolutionary tree where my cursor is here. VHL. That's a very earlier event present in every tumour cell, but then you've got mutations that drive tumour progression. Don't worry about what these genes do, but just accept it that if you don't mind that these are driver genes as well, these are driving progression in some cells but not others. So what we concluded from this is that biopsying a 10 by 5 by 5 centimetre tumour and telling a clinician we understand how this tumour behaves is a little bit like me telling you that I could map let's say, an area of huge ecological diversity between Kenya and Tanzania, from the Masai Mara down to the Ngorongoro crater, and give you a corridor that is 250 kilometers from north to south, measuring 1 meter by 1 meter by 1 meter. And I could take 10 meters of that 250 kilometer corridor and map the ecological diversity in that corridor and tell you I know everything about that stretch of road from Masai Mara down to the Ngorongoro crater. You would look at me and think I'm utterly insane. But that's actually what we're doing, sequencing a single biopsy. We're taking a very small region of a very large primary tumour and telling something about that diversity of the ecological environment and the growing tumour mass and mapping it onto the disease as a whole. We clearly can only map a very small part of that patient's disease from a biopsy from a patient's tumour. So what does this tell us about drug response? So we gave this patient a drug that targets a particular protein in this patient's cancer called mTOR. Don't worry about what it does, it's a survival pathway. We've got a specific class of drug called an mTOR inhibitor, which we can give this patient. When we took the tumour out, we can then map the pathway to see if the mTOR pathway is switched on or switched off, which we did. And this is what we found. So we found, I'm afraid I haven't got I'll use the cursor on the keyboard. We found that most of the regions of the tumour had the pathway switched on, the mTOR pathway switched on, shown in green here. And that maps to region 5 by immunohistochemistry. This brown here means the traffic light is on, it's green. The drug is not working. In contrast, regions 4 of the tumour and the metastatic sites had the mTOR pathway switched off. There's not much brown here, so the traffic light is red. So there's heterogeneity of drug response, diversity of drug response. We've got the drug working in some parts of the tumour, but not others. So we wanted to know why that was. And what we found is when we looked at the sequence of the tumour in the different regions, we found this activating mutation in the mTOR kinase. So this protein has got a mutation in some parts of the tumour where the pathway is switched on, but not others, where the pathway is efficiently switched off by the cancer drug. And this particular mutation makes that pathway constitutively active. We can't switch it off with the drug that we have in our hands that we offer the patient. So this shows that diversity really does matter. Genetic diversity means that you get heterogeneity of drug response. So the question the reviewers asked us, I remember being on holiday at the time when we got the referees' reports back and they rejected the paper outright because it was just one patient. They said, well, okay, it's all very well. You're saying that cancer diversity is occurring in all tumours, but you've only shown us one case. So the question they asked us is linear evolution the norm and you've just found an outlier? Or is branched evolution the norm and is linear evolution just rubbish? And so we then, over a course of the next 2 years, sequenced 10 patients. Every single tumour shows branched evolution. And in fact, we've not really found, since then, having sequenced hundreds of tumours, evidence for linear evolution at all. That branched evolution is the norm. Where we do see linear evolution, I think that's sampling error. And I think we have to just understand that branched evolution is what we're facing when dealing with cancers in the clinical setting. So what are we going to do about it? It's all very well describing this. We've got to actually do something about it. And I think the first thing is that we need to think about tumours like trees. We've got trunk events that are present in every tumour cell. These are the events we need to target and target mercilessly. And then we've got branched events that are present in some cells but not others. And that's what's going to drive resistance to therapy, a bit like that mTOR mutation I showed you earlier. Mutations that are present in some cells but not others drive resistance to therapy. And we need to understand the clonal architecture of the tumour. And I think this all has some impact on the challenges that we face treating patients with lung cancer. It probably tells us to some large extent how tumours acquire resistance to therapy, how they acquire drug resistance. It tells us why we found it very difficult to predict outcomes for cancer patients, so-called biomarkers that allow us to either predict drug response or predict pathways of resistance or predict prognosis or outcome in general, because we're only sampling a very small part of the tumour from a single biopsy. And I think the most important part of this is it really tells us why the cost of cancer care is so expensive, because drugs almost always fail in late-stage disease because of this diversity in this ever-evolving tumour. And it's worth saying that in a tumour there can be between 1 and 10 trillion tumour cells. That's 10 times more, or 10 to 100 times more tumour cells than there are stars in our galaxy in one patient. This is an enormous amount of diversity that is apparent in a patient with late-stage metastatic disease. So what are we going to do about it? Well, we know that evolution is very constrained, and what I want to get across in my lecture today is the fact that that some very, very important people in evolutionary biology have described this problem of branched evolution, or this concept of branched evolution, way before me. And as you walked into this Darwin lecture theatre today, you saw Darwin's I think diagram that he drew in 1837 that first postulated branched evolution. So really none of what I'm describing today is really very original. Most of what I'm describing today is simply redescribing evolution as it has been described in the last 157 years since Origin of Species was published in tumours. So we want to exploit constraints to cancer evolution, and in a phenomenon we call parallel evolution, or that is known as parallel evolution. What do I mean by that? Well, it's essentially different subclones in an evolving tumour acquiring the same characteristics over the disease course. So this is shown here in the first patient we sequenced, where we see multiple different mutations occurring in a tumour suppressor gene called SETD2 occurring in different regions of the patient's tumour. But they're distinct mutations, so they're being acquired at multiple different times during evolution and selected for. And that's called parallel evolution. And that tells us that there's huge constraints to evolution that we might be able to exploit. In addition, that parallel evolution can not only affect the same gene in different ways, but it can affect the same protein complex. This protein complex here, called a SWI/SNF complex, affects the ability of cancer cells to alter the way in which they transcribe genes into proteins. And it can be mutated in many different ways. Because it's a large complex, you can mutate multiple different parts of that protein complex. I think there are about 6 or 7 different protein components that form that complex. You can mutate each different protein in many different ways to essentially converge on the same ends, namely disruption of that complex. That's shown here. In different regions of the tumour, you have mutations in PBRM1 or ARID1A or SMARCA4-BRG, essentially different ways of disrupting the same complex that gets to the same end. Now, of course, evolutionary biologists described this 157 years ago. Charles Darwin's Origin of Species was published 157 years ago to the day yesterday. And he described this in his book. He said, the common rule throughout nature is infinite diversity of structure for gaining the same end. And essentially what we've been doing over the last 200 years is redescribing what Charles Darwin first described then. Conway Morris did it in 2003. Describing the recurrent tendency of biological organization to arrive at the same solution. That's convergent evolution in species, parallel evolution as we see it in tumours. So I've naively been running a lab thinking that we can predict cancer evolution and we can use that as a tool to offer patients a therapy to stop tumours going down a distinct evolutionary route. Now, Charles Darwin had the answer to this back in 1859. He said basically what we're trying to do is impossible, because he postulated this problem when he's trying to predict evolution, throwing up a handful of feathers and all fall to the ground according to definite laws. But how simple is the problem where each shall fall compared to that of the action and reaction of innumerable plants and animals which have determined in the course of centuries the proportional numbers and kinds of trees now growing. So he was pondering this problem of evolution and whether it was predictable. And I think you can tell from what he's saying here that he doesn't think it is predictable. We're hoping that evolution in a patient is predictable, but maybe we too will fail. But we'll try nevertheless. So that set up really the background to what is now called the TRACERx Consortium, which is a national consortium with about 15 hospital sites across the United Kingdom to try to understand cancer evolution in intense detail. And we're focusing on lung cancer, and we're focusing on early stage lung cancer from diagnosis through to death. And the idea is that we are multi-region sequencing 842 patients with non-small cell lung cancer and understanding how their disease evolves and adapts through treatment and ultimately in about half of cases, sadly, to death. Because we need to understand where the lethal subclone is coming from and how it's seeding recurrence and ultimately death at metastatic sites. And we have an autopsy program called PEACE that patients we hope will consent to, and indeed some already have, that will be able to acquire multiple metastatic samples to understand how that evolved from the primary tumour. And the endpoint of the study is to understand, is there a relationship between the shape of the tumour phylogenetic tree and outcome? So are the more complex baobab-like trees associated with worse outcome than more simple palm tree-like tumours that have long trunks and short stubby branches that don't have very much diversity? This is the cohort. We've got about 100 patients so far of our 842. We're about, in analysis terms, about an eighth of the way through the study. We've recruited about 468 patients, I think, now who've kindly donated their primary tumours to us for analysis. And unfortunately, the analysis lags behind the recruitment. So what I want to show you now is some early data from that study, unpublished data, of really how diversity is created in a lung cancer tumour. Now, it turns out we think that diversity is important for evolution, and it's hardwired during the course of evolution as part of the way in which that tumour develops robustness and fitness. And I'll try and allude to that in a minute or two. And the three processes that guide tumour evolution in lung cancer are these three processes. Aging, an aging signature, which is referred to as spontaneous deamination. This is a background mutation rate that all of us are subjected to every second of our lives. There's nothing we can do about this. A second process is smoking. There's quite a lot we can do about that, obviously, and that's essentially these G to T or C to A mutations that occur in the cancer genome, induced by tobacco carcinogenesis. And then there's this third mutational process called APOBEC. Try not to worry too much about the jargon, but APOBEC is essentially an enzyme, a protein in one's cells that fights off viruses. It's switched on in response to viral infection to mutate viral DNA. Now the problem is that our own tumour cells do the same thing. They switch on APOBEC to induce mutations. Now whether there is a cause and consequence, there's clearly no anthropomorphic view of this, but this is a process that is selected for during tumour evolution, as I'll show you in a minute or two. And it drives mutations by mutating cytosines to uracils, essentially mutating DNA. Now, we can look at our 100 patients and ask what type of mutations we're seeing across the genome. You see these grey mutations. Those grey mutations are smoking mutations. So we're looking at the burden of the mutations on the y-axis, and on the x-axis is one individual patient. Each bar is one individual tumour. And we're looking at the breakdown of the mutations according to the mutational type. So the overwhelming mutational type is the smoking signature, the C to A mutations. But you also see a lot of orange and yellow mutations. These are APOBEC mutations. So we can now ask a question, when do these mutations occur? And the smoking mutations are essentially the trunk mutations. So almost all of the trunk mutations are C to A smoking mutations. So these are occurring early on in tumorigenesis. These are the initiating mutations. So cigarette smoke is pretty important in driving lung carcinogenesis. I don't need to tell the audience that, but I think this is pretty clear evidence that these early mutations, truncal mutations, are smoking induced. What about the branch mutations? Well, the branch mutations are driven, we think, by two types of mutations. Aging, which is essentially a reflection of cell division, and as cancers grow, they proliferate faster. So because they're proliferating faster, they can age faster and they develop more aging mutations. But also this endogenous, this human-encoded enzyme, APOBEC, is driving branch mutagenesis. So it's driving branch mutations. And even in a current smoker who's smoking 30 to 40 cigarettes a day, this will be the dominant mutational force. APOBEC will be the dominant mutational force in that patient's tumour very often. So going back to that point, is linear evolution or branched evolution the norm? This is just the evidence really that I can show you that the overwhelming majority of tumours show branched evolution. This is 100 patients and the different trunks and the branches. Now what we're looking at here is in the branches the different types of mutational processes. And I show you this because I think it's important, because I sort of naively hope we could target evolution by inhibiting APOBEC or perhaps stopping smoking-related mutagenesis. But what you see here is that the different branches have different colours, and that shows you that in different tumour regions you've got different mutational processes driving evolution. So you have APOBEC in one branch and smoking in another, or aging in another and APOBEC in another. And so if we had a drug that could target APOBEC or one stop smoking, you'd spring up another branch probably by virtue of APOBEC-induced mutagenesis. Nature will find a way. Nature will find a way of driving diversity, and APOBEC is important in doing that. So I've talked a lot about microevolution, single point mutations driving evolution. This is sort of the Darwinian approach to evolution. Single point mutations over time, slowly changing the behavior of the cancer cell. But there's more to the story than that. And there's this evolutionary biologist who I think is a bit of a star called Goldschmidt, who wrote in the 60s that he was going to be considerably more famous in 40 years' time than he was in 1959 when he published this book, Material Basis of Evolution. And he was a bit pompous, and this rather put people off him. And he coined this phrase, hopeful monsters, which didn't help. Because he postulated that evolution was driven by rearrangements of what he called the serial chemical constituents of the chromosome. That is, chromosomes breaking up and fusing into random orders that would occasionally create a hopeful monster or a whole new species. Now the problem is that one has some difficulty thinking about this in the context of a sexually reproducing organism. But in the context of cancer, this makes a lot of sense. Because these types of chromosomal rearrangements that he showed in figure 35 of his book is what we see in cancer writ large. And I just want to focus on this beautiful paper that was published in 2014 because I think it illustrates just the scale at which chromosomal rearrangements can alter the way in which species evolve. So this beautiful species, the gibbon, is unusual and has somewhere between 19 and 26 pairs of chromosomes, unlike other ape species, which have a very stable chromosome content of 24. Now, The gibbon diverged from all ape species about 4 million years ago during a time of very rapid evolutionary change in the environment where sea levels were changing and species were becoming isolated, beautiful conditions for speciation. So this group sequenced the gibbon genome. Now, they did that because they're rather obsessed with a gibbon for the simple reason they're like us in many ways, in that they walk upright and they don't have tails, and like some of us, they're monogamous. But they are also operatic sopranos. They're extremely sociable animals. And very different from many other ape species. So they're obsessed, this group were very keen to understand a lot more about how this beautiful species diverged from other apes. And what they found is that in this genome, about 33 times, a retrovirus element had hopped and skipped and jumped into the genome multiple different times around genes that were involved in chromosome segregation at cell division, and probably had disrupted the karyotype or the chromosomes to such an extent they'd led to this wide array of chromosome makeup in this ape species that allowed diversification of the ape from its ancestors. And so I think this shows to some extent how the reordering of the serial chemical constituents of the chromosome can help a new species evolve. Now, if that can generate a new species, what's happening in cancer? And this is what happens in cancer. We see a wide array of different chromosomal makeups in different tumor cells within one patient. We can see aberrations in number, in structure, and actually, most importantly, we see this problem of whole genome doubling events. Now, whole genome doubling is important because us and others have found that whole genome doubling events in cancer can lead to the rapid onset of cancer evolution. Cancer cells evolve much faster when they've doubled their genome. Cancer cells can metastasize. It's a high-risk feature of tumor genomes if they've doubled their genome. And cancer cells in mice grow much more rapidly if they've doubled their genome compared to their diploid counterparts. This is a high-risk feature. But like many evolutionary biologists before us, this has been well described in the literature. A lot of the foodstuffs, the plant foodstuffs and vegetables we eat have doubled their genomes, and they've done it for a reason, to adapt to harsh environments. And Julian Huxley, the Nobel Prize winner, described in Evolution, the Modern Synthesis, that tetraploid forms, these are genome doubled forms, when he's referring to nature and ecology, in many cases are more generally vigorous, a fact reflecting their distribution which is frequently wider than that of the diploid variety. I think one can now begin to see how tumor cells, when they've doubled their genome, metastasize. One thinks about it like this, or a plant colonizing arid deserts, one can begin to see why doubling a cancer genome is so good for a cancer. It can now explore other sites, the lung, the liver, the bone, or the brain that it's not normally used to, and colonize and express new genes, or increase expression of those same genes, but just a higher dosage to be able to adapt to a new environment. And in lung cancer, this happens a lot. 80% of our early stage lung cancers have doubled their genomes very early on in tumor evolution. It's a clonal event. It's probably occurring before that first tumor cell invades through the basement membrane and spreads either within the lung or throughout the body. This just shows, I'm afraid it's a bit of a messy plot, but for the most part, genome doubling is a very early event in tumor evolution in lung cancer. I also want to just quickly emphasize how rearrangements of the genome are important in an asexually reproducing organism like a cancer, because it can help them rewire their evolutionary histories. I'll just show that in cartoon format here. If you think about an evolving tumor clone, you can do one of two things. You can, by creating chromosomal instability, you can lose whole segments of the genome that enable you to lose early events in tumor evolution that you may no longer want, that are no longer hardwired into your genome. You know, let's say you've got a gene that helps with invasion, but later on exposes that tumor cell to the immune system. You may want to lose it. The tumor cell may want to lose it, and that's what they do. They lose chromosomes to rewire their evolutionary histories. They lose parts of chromosomes, small segments of the chromosome, or small, just, individual nucleotides. They've got this huge capacity to rewire their evolutionary histories. And this, we found, is also a driver of parallel evolution at the chromosomal level in lung cancer. Tumours can gain oncogenes later on in tumour evolution from either the paternal or the maternal chromosome in different subclones of the same tumour. Nature will find a way. And so back to the question of what's really guiding outcome. Is it heterogeneity of point mutations, or is it chromosomes, parts of chromosomes, as Goldschmidt, that evolutionary biologist, would lead us to believe? So we can now test this, because there's a prospective study in TRACERx to look to see what the relationship of point mutational heterogeneity against chromosomal instability is to clinical outcome. And these are the data. There's no relationship between microevolution, point mutational heterogeneity, and outcome. There's no relationship that we can see, at least in the first 100 patients. But there's an incredibly strong relationship between the degree of chromosomal instability and outcome. That tumours that are chromosomally stable, patients do extremely well. Patients that have very chromosomally unstable tumours do very badly, and that appears to be independent of disease stage. So even within stage 1 or stage 2 cancers, this holds. So chromosomal instability, we think, is important. And as Goldschmidt said in his book, Material-Basis of Evolution, when referring to large-scale genetic changes, such a mutation need only be an event of the most extraordinary rarity to provide the world with the important material for evolution. And I think the emphasis here is it's low-frequency events that can be large-scale that influence outcome. And that's why predicting outcome in cancer is so difficult, because ultimately it's a single subclone from a heterogeneous tumour that seeds the metastatic site that ultimately ends up resulting in death for that poor patient. So predicting the future is difficult. So what are we going to do about it? Well, I just want to show you some ideas that we're working towards now about how we're going to manage heterogeneity. This is what we've been doing over the course of the last decade, targeting one point mutation in the tumour's evolutionary tree. So I can just show you this video. This is Charles Darwin. His I think diagram is here that he drew in 1837. This is how tumours evolved too. So they evolve from a single cell in the trunk that ultimately develops diversity, cells that have new mutations that pop up in the branches. Some die, some are productive, some go on to spawn metastasis, some go on to spawn drug resistance. And the problem is the cancer drugs we've been using only ever target one clonal alteration present in the trunk of the tumour's evolutionary tree. And in a tumour with 10 trillion tumour cells, there's always going to be one or more tumour cells before a cancer doctor has offered a patient a drug that will be resistant to therapy, to that therapy we're using, and a branch will grow out and allow the tumour to become resistant to treatment within 3 to 12 months of therapy, resulting in the huge cost of therapy that we see in the clinic and failure of treatment and ultimately death from cancer. So what we need to be doing, the evolutionary biologists tell us, is targeting 3 or more truncal alterations, multiple truncal alterations, so we limit the possibility there is one star in that galaxy, one tumour cell in that patient that is resistant to treatment. That's all you need, one tumour cell out of 1 trillion that's resistant to therapy. And so when one thinks about it, this is what we're trying to do. We've got this heterogeneous tumour with resistance developing. We want to target multiple clonal alterations, that's multiple truncal alterations, 4 or more, hopefully, multiple, that limit the ability of that cancer to develop resistance to therapy and ultimately resist treatment. So if we can shrink the tumour, hit the tumour down at the trunk, we hope to be able to tackle all of the branches simultaneously, wherever they are, in the lung, the liver, or the bone. And so what we've seen is this huge opportunity in TRACERx in lung cancer, that the smoking cancer genomes have very long trunks. So what we found is if you compare a smoking cancer genome here on the left to a non-smoking cancer genome on the right, and you look at the length, the size of the trunks and the branches are not dissimilar, but the trunks are much longer in a smoking genome. So there's somewhere between 5 and 10 times longer in a smoking cancer genome than a non-smoking cancer genome. That means you've got 5 to 10 times more mutations that the immune system might be able to see. Every time you get a point mutation, there's a risk that an immune cell in your body's tumour, in your body, will see that tumour and might be able to tackle it. So we've now got a pipeline set up between the Francis Crick Institute and University College London and the hospital to multi-region sequence tumours now on TRACERx to give us an idea of what those truncal alterations are. And we've now got new technologies that allow us to extract T cells, immune cells, from the patient's own tumour that specifically recognise those truncal alterations. And we can do that multiple times, not just identify one T cell, but in fact we've got one patient now, we've got 6 different T cells, immune cells, that we found in peripheral blood that recognise truncal alterations that we hope we can liven up and ultimately infuse back into the patient to hit the patient multiple times in the trunk. So what does that look like in cartoon format? It looks a bit like this. You've got this growing heterogeneous tumour, you've got the immune cells that are circling it that are confused by the growing diversity as the tumour's evolving. that are put off by the diversity because they're simply seeing the branches, not the truncal alterations. But we know that even complex regions of the tumour, shown in blue, purple and green, bear hallmarks of their origins in yellow. These are the trunk clonal alterations. And we've found now rare immune cells that specifically recognize those yellow truncal alterations and multiple immune cells that recognize these truncal alterations. So essentially the body's producing its own immune cells that are acting like a mini pharmaceutical company, developing a bespoke therapy for each patient in one's own body. The problem is those immune cells need a boost because they're put to sleep by the tumor in a predator-prey relationship. And so what we hope to do is expand, isolate these immune cells through these genomics approaches I've been telling you about, expand them up outside the patient, ultimately give them back to the patient in much larger numbers to overwhelm the tumor, or develop vaccination approaches that specifically target those truncal antigens that I've been talking about, those truncal mutations. So that sort of pretty much summarizes my talk, that heterogeneity in branch evolution is the norm, that heterogeneity is here, we've got to deal with it. We can't just continue targeting single point mutations because resistance is inevitable. I think it's clear that there are genome doubling is important in evolution. APOBEC, this mutational mechanism, is a driving force of branch evolution, and I hope that this burden of clonal alterations, truncal alterations in these tobacco-associated cancer genomes will be an Achilles heel for cancer therapies in the future and will hopefully enable us to improve outcomes for patients suffering from lung cancer. I just want to thank all the people who've made this possible, Cancer Research UK, the Rose Trees Trust, ERC, Royal Society, Medical Research Council, all the people who've helped fund this, as well as University College London and the Biomedical Research Centre, all the people in my lab, principally shown in red, who've collaborated and who've worked hard to produce the data. This is team science at quite a large scale, so I present this work really on behalf of the TracerX consortium across the UK. Thank you all very much for listening.
Thank you. We have 5 minutes. One question? We just have time for one question. Okay. Yes, I saw you first.
I'm reminded of treatment for HIV.
Yeah. Yeah. So, yeah.
Hi. So I'm reminded of treatment of HIV, where you have huge diversity in the population, and clinicians treat with multiple drugs to try and prevent resistance. Is that something that you foresee in this case as well, treating with multiple different types of strategy?
So we have to target multiple truncal events. It's a very good question. The answer to your question is you've got to bear in mind the difference between HIV and cancer is in cancer we're treating a disease that has evolved from man 6 to 12 months ago, possibly longer, some cases 5 to 10 years ago. HIV evolved from man billions of years ago. So every enzyme we target in HIV does not have a homologue in the human genome. So when you treat with a drug, you've got a much greater therapeutic window, so you can combine multiple drugs to eradicate the virus. Whereas when you combine multiple drugs in cancer, you're essentially exploiting a very narrow therapeutic window between self and non-self. And so you can't dose escalate. You end up reducing the doses of the drugs because you get essentially on-target effects on normal tissue. So part of the problem with a lot of drugs we use, like EGFR inhibitors in lung cancer, is you get terrible skin rash, diarrhea, and when you combine with another drug, patients just can't tolerate it. So we can't exploit that therapeutic window well enough, unfortunately, to combine drugs. Whereas if we have immune cells, or your immune system has huge specificity for mutations, so we can combine multiple immune cells together, we hope, because you're only targeting one-point mutations, not anywhere else in the body. So that's huge specificity, we hope. So combination therapy is definitely the answer, but it needs to be absolutely 100% specific.
I'm sorry, but we're going to have to finish here. There is a class coming now. You can always email your questions to Charlie or to UCL. Thank you.